Welcome to this lecture on cell biology. Today, we are going to explore a fascinating and crucial topic, cell-cell junctions. If you have ever wondered how trillions of individual cells in your body manage to stick together, communicate, and function as unified tissues without falling apart, you are in the right place. We will look at the biological seals, the mechanical anchors, and the communication highways that make complex life possible. Let us dive in and see how cells build our bodies. To begin our journey, let us look at tight junctions, which act as the biological seals of our tissues. Imagine building a brick wall. Without mortar, water would just seep through the cracks. In our bodies, tight junctions are that mortar. They are formed by specialized proteins called claudins and occludins that pull neighboring cell membranes incredibly close together. This blocks the paracellular pathway, meaning molecules cannot just slip between the cells. Why does this matter? Without tight junctions, highly acidic stomach juices would leak out and digest your own organs, and harmful toxins could easily cross the blood-brain barrier. They give tissues absolute control over what enters and exits. Now that we understand what tight junctions do, let us look at how scientists actually measure their strength in a laboratory. We use a formula for transepithelial electrical resistance, or RT. Conceptually, this is based on Ohm's law, but adapted for a biological tissue. The formula is RT equals V divided by I multiplied by A. Here, V is the voltage difference across the cell layer. I is the electrical current applied, and A is the surface area of the tissue. If the tight junctions are forming a strong, watertight seal, it becomes very hard for electrical ions to pass through. Therefore, as the seal gets tighter, the resistance, RT, increases significantly. Before we move on, let us see this formula in action with a concrete example. A scientist is testing intestinal cells with a surface area of 2 square centimeters. She applies a current of 0.0005 amperes and measures a voltage drop of 0.05 volts. We need to calculate the resistance to check the integrity of the barrier. First, we divide the voltage by the current, which gives us the raw resistance of 100 ohms. Then we multiply by the area of 2 to standardize the measurement. This gives us a final transepithelial electrical resistance of 200 ohm square centimeters. This high value tells us the cells have formed a healthy, secure seal. Having calculated the resistance, let us visualize exactly what is happening at the microscopic level. In this diagram, you are looking at the membranes of 2 adjacent cells. Notice the apical side at the top and the basolateral side at the bottom. Right in the middle, you can see the claudin and occludin proteins acting like a Ziploc track, physically stitching the 2 membranes together. See the arrow representing water and ions trying to sneak down between the cells? It is completely blocked by the tight junction. This visual perfectly illustrates how these structures force molecules to pass through the cell itself rather than leaking around it, maintaining strict compartmentalization. While tight junctions seal cells, they are not designed to handle heavy physical pulling. This brings us to our second type of connection, adherens junctions, the mechanical anchors of the cell. Think of a team of mountain climbers tied together with heavy-duty ropes. If one slips, the rope distributes the force so nobody falls. Adherens junctions do exactly this for your tissues. They use proteins called cadherins that reach across the space between cells and link up. Inside the cell, these cadherins are firmly anchored to the cell's internal skeleton using linking proteins called catenins. This is why you can pinch and pull your skin without it ripping apart. The adherence junctions distribute that mechanical stress across the entire tissue. Now that we know adherence junctions handle physical forces, let us look at the mathematical relationship that defines this mechanical stress. The formula is sigma equals F divided by AJ. Here, the Greek letter sigma represents the mechanical stress experienced by the tissue. F is the total pulling force applied to the cells, and AJ is the total cross-sectional area of the adherence junctions connecting them. Conceptually, this tells us that the stress on any single junction decreases if the area or number of junctions increases. Conversely, if the pulling force F increases without enough junctions to support it, the stress sigma goes up, which could eventually cause the tissue to rupture. Let us apply this stress formula to a real-world scenario involving skin cells. Imagine a patch of skin is subjected to a stretching force of 0.004 newtons. The total area of the adherence junctions holding these cells together is 0.00002 square meters. We need to calculate the mechanical stress to see if the force is safely distributed. By plugging our values into the formula, we divide the force by the junction area. This gives us a final mechanical stress of 200 pascals. Because the adherence junctions spread the load effectively, this stress remains at a manageable level, ensuring the skin cells do not tear apart from one another. To truly appreciate how this stress is managed, let us examine this diagram of an adherence junction under tension. You can see cell 1 on the left and cell 2 on the right. In the intercellular space between them, the blue cadherin proteins are interlocked, holding tight. But the most important part is inside the cells. Notice how those cadherins are directly attached to the red actin filaments of the cytoskeleton. When the outward pulling forces, shown by the black arrows, stretch the tissue, the tension is transferred directly into the structural framework of the cells. This diagram perfectly captures how adherence junctions act as biological safety ropes, keeping our cellular communities securely fastened under physical strain. We have seen how cells seal themselves and anchor together. Now, this brings us to our third and final type, gap junctions, the communication highways of the cellular world. Think of two adjacent hotel rooms. If you open the connecting door between them, people can easily pass back and forth. Gap junctions are exactly like those connecting doors. They are formed when two half channels, called connexons, align and dock across the space between cells. This creates a physical hydrophilic tunnel that directly connects the cytoplasm of both cells. This allows ions and small molecules to flow freely without ever entering the outside fluid. In organs like your heart, this direct communication allows millions of cells to share electrical signals and contract at the exact same fraction of a second. Because gap junctions transmit electrical signals via ions, we can quantify their communication capacity using the total electrical conductance formula. The formula is GT equals N times PO times gamma. Let us break down these symbols. GT is the total electrical conductance between the two cells. N is the total number of gap junction channels present. PO is the probability that any given channel is actually open, which is a fraction between 0 and 1. Finally, the Greek letter gamma represents the conductance of just one single channel. Conceptually, this shows how cells control their communication. To increase the signal flow, a cell can either build more channels to increase N, or chemically signal existing channels to stay open longer, increasing PO. Let us see how this conductance formula applies to a beating heart. Imagine two adjacent cardiac muscle cells connected by 5,000 gap junction channels. The single channel conductance is 100 picosiemens, and the probability of the channels being open during a heartbeat is 0.8. We want to find the total conductance to understand how easily the heartbeat signal can travel. We plug our values into the formula. First, multiplying 5,000 channels by the 0.8 probability tells us that 4,000 channels are effectively open. We then multiply that by the single channel conductance of 100. This gives us a final total conductance of 400,000 picosiemens. This massive electrical capacity ensures the heartbeat signal passes almost instantaneously. To wrap up our understanding of this communication highway, let us look at the structure of a gap junction channel. In this diagram, you can clearly see the membranes of two neighboring cells. Spanning across the gap between them is the complete gap junction channel. Notice how it is made of two halves, the connexons, which are each built from 6 individual connexin subunits. When they dock together, they form that central pore you see in the middle. The arrow passing straight through the pore represents the rapid flow of ions and small molecules directly from one cytoplasm to the next. By regulating these specific channels, cells can finely tune how closely they interact, transforming isolated cells into a unified functional network. We have covered a lot of ground today, so let us summarize the 3 most important ideas about cell-cell junctions. First, tight junctions are the biological seals. They block the spaces between cells to maintain strict internal compartments. Second, adherens junctions are the mechanical anchors. They tie the cytoskeletons of neighboring cells together to distribute physical stress and prevent tearing. Finally, gap junctions are the communication highways. They create direct tunnels for rapid ion exchange, allowing tissues like the heart to synchronize perfectly. Together, these 3 distinct types of junctions are what allow billions of isolated, individual cells to come together and form the resilient, unified tissues that make up your body. Thank you for joining me in this lecture.